Thank you for being here to hear this talk. I'm going to be covering a lot of ground. I've only got 14 slides to show, so that means I have to do them in less than 2 minutes apiece. So a lot of the areas of neurotransmitters I'm going to gloss over. Some of them I'm going to deal with in depth. And I'm going to try to not just cover issues of the biochemistry, but the issues of what does this mean to you in terms of your consciousness, your perception of the world, your ability to think, your ability to emote, to accomplish work, to be happy in your life, which is a big jump, because scientists can investigate, biochemistry, because you're dealing with little parts. So it's like a computer chip in terms of having switches and electrons that move and bits of data, ones and zeros. But the human brain doesn't work that way. If you have a nerve in your leg and you stimulate it with a signal, so a nerve impulse travels up the neuron, you do not perceive that as hot or cold or pain or touch or pressure or anything like that. Okay, so the issue of, for example, sensory input depends more on the rate of the signal. So if the nerve is firing once a second, you don't feel anything. But if it fires twice a second, then you start to feel something. Or if it slows down to 1 every 2 seconds, you feel something. So it's there's no direct linear thing between bits in the nervous system and your subjective consciousness like there is between a computer chip and your memory and what the computer is doing. Okay, basic question about neurotransmitters. What are neurotransmitters? Well, neurotransmitters are chemical signaling agents, like words. If I say serotonin, that's a word. It comes out of my mouth, I would be the broadcasting cell, and goes into your ear, you're the target cell or the postsynaptic cell, and you hear this word and it means something to you. Now, if we look at all the people in the audience, we can say, well, not everybody's ears are necessarily going to function the same. So some people may need hearing aids where the signal gets turned up in order to function normally. Other people may be distracted by something and may miss a phrase or a word here and there, and therefore not get part of the message. Likewise, if you don't speak English as your primary language, it may be more difficult to understand what's going on. And so we find a lot of aspects of neurotransmitters that can be made analogous to like verbal communication in terms of all the variations that you can see in communication. So sometimes, for example, not only do I say a word, but I'm also listening to the word. So I hear myself say something. Now, a lot of speakers don't. If you've been to college, professors, sometimes they just stand up and profess. It's like they're broadcasting to the audience and you're supposed to be a sponge. And they don't really pay much attention to whether or not you're getting something, or they don't even necessarily hear what they're saying. But so, for example, let me go on to here. Now, here's the sending cell. This is like me. And here's the postsynaptic cell, which is hearing the signal. The neurotransmitter, in this case, we have norepinephrine, which is being released and is being absorbed by a postsynaptic receptor, which would be analogous to your ear. Okay, now, that postsynaptic receptor, I'll get to this later in the talk, will produce a metabolic change inside the cell. That's the issue of your brain paying attention to what's going on in your ear. But we have, this is my ear, okay? So this is an autoreceptor. This is a receptor just like the postsynaptic receptor, but it's on the presynaptic side, which is picking up the neurotransmitter that's released and then reacting to it and sending a signal back, which causes a change in the sending cell. So a sending cell can become sensitized to become more louder and louder and louder in terms of how it's talking to its neighbor, or it can become quieter and quieter and quieter. It can release less neurotransmitter or more neurotransmitter. The reuptake receptor picks up neurotransmitter and sends it back in. That can also provide a control signal to regulate something inside the cell, or it can also just conserve material. It's a recycling mechanism. So how many people have heard of serotonin reuptake inhibiting drugs? Prozac, Zoloft, Paxil, that kind of stuff? That's what this drug does, is it interferes with this particular enzyme here so that the neurotransmitter stays outside in the space and less of it is available inside the cell, being stored to be released at some future point. The actual mechanism, in the case of norepinephrine, there are these things called vesicles, which are little tiny membranes that surround a sac. And these are filled with a neurotransmitter or multiple neurotransmitters, or a neurotransmitter and something called a neuromodulator. This membrane sits in the cell, and then there's a mechanism that fuses it with the outside membrane, which then creates a hole, and then the neurotransmitter leaks out and travels across this gap there, which is called a synaptic gap. This particular kind of neurotransmission is like two people talking face to face. But we have other kinds of neurotransmitters as well that are broadcast, like me talking to this entire group through a microphone system. For example, nitric oxide that controls vascular muscle relaxation and, for example, in men, erections. So that would be an example of a neurotransmitter that's released by one cell, and it diffuses as a gas to many cells, not only that are immediately adjacent to the cell that's signaling, but several cells away. So neurotransmitters is not necessarily just this kind of, two surfaces touching up against each other. It can also be long distance. Nitric oxide is one example of that. Carbon monoxide is another gaseous neurotransmitter. This is an example of one particular neurotransmitter called serotonin, which is a key one to human beings for their well-being, because this helps us sleep, and it helps us handle pain. It helps us be calm and tranquil, and it keeps our blood pressure down. It also makes us pleasant towards each other. So this particular neurotransmitter here is called serotonin, which is this pool right down here. The reuptake is drawn as an arrow that recycles back on itself. And there's a catabolic pathway that destroys it. And then there's a synthetic pathway that feeds it. And there are two substances, tryptophan, which is an essential amino acid, and 5-hydroxytryptophan, which is its immediate metabolite, that can also be taken supplementally. So actually right now, 5-HTP is more commercially readily available than tryptophan is due to FDA politics. But it's easy to understand the basic concept that in order to modify the level of serotonin, you want to input additional material into the upside of this pathway so that tryptophan flows into 5-hydroxytryptophan and 5-HTP flows into serotonin. Make sense? Feeding the system. We also have here drawn a negative feedback control loop here, which responds to serotonin that goes back up and rate limits this enzyme, tryptophan hydroxylase, so that if you supplement tryptophan and you make serotonin, it shuts off the flow of tryptophan. So this has some disadvantages in that you don't get as much effect as you might necessarily want, but it also has some benefits in terms of safety. You can't really produce serotonin system, overload this serotonin pool by tryptophan. It's extremely hard to do. If you combine tryptophan with Prozac, you can do it. If you combine tryptophan with an MAO inhibitor, you can do it. But by itself, tryptophan is nicely controlled by this feedback loop. So 5-hydroxytryptophan, because it's past that bottleneck, is not regulated by that feedback control loop. So, yes?
What happens if you do overload it? What are the symptoms?
I've never experienced them myself, and I don't want to. But people feel really bad. The hair on the back of your neck stands up. You can have blood pressure problems, feelings of anxiety, impending doom. It's from what I've been told, extremely unpleasant. And I believe it can be fatal in extreme circumstances. Gary, can you describe it more than that?
No, I think that's pretty good.
Okay. Over on the side here, we have the actual chemical structures: tryptophan, 5-hydroxytryptophan, serotonin, N-acetylserotonin, and melatonin. So this shows a kind of flow diagram of how an actual molecule, an actual substance, is being changed. So we have the introduction of this hydroxy group here. We then have the removal of this carboxy group from to here, which means this nitrogen becomes a sole function. This is an amino acid, amino and acid, amino and acid. So both of these are amino acids, and this is called an amine or a monoamine, which monoamine oxidase metabolizes. So yeah, I'm sure you really wanted to know that. But the idea is that you have to have serotonin production in order to make melatonin, and so these materials flow down. So it's like an automobile factory where you put in a bunch of parts at one end, and a machine comes down and stamps this, and then bends it here, and then welds it here, and then paints it there, and eventually at the far end you get a car or a radio or something. Here's another neurotransmitter system. This is acetylcholine. This is a very powerful neurotransmitter system, and this is involved in intelligence, memory function, as well as motor control, muscle tension, coordination, dexterity. So it's a high-level brain function and a lower brain motor control neurotransmitter. Also regulates peripheral neurotransmitter systems relating to things like gut motility. So if your cholinergic system is very sluggish, you can get constipation, for example. Or if it becomes overly active, diarrhea. So we have here a series of reactions where you can see the red arrows that show the flow of material. So choline, which is part of acetylcholine, has the nitrogen with the 3 methyl groups on it and the positive charge. And then it has the OH group here, which is, in this case, acetylated versus acetylcholine. So by attaching an acetyl group to it, you get acetylcholine. Dimethylaminoethanol it has 1 less methyl group, so it's not charged. And this has an advantage that I'll display later, that choline is rejected by the blood-brain barrier, so when you take it orally, it tends to feed your body and not your brain, whereas DMAE feeds both. Down here we have phosphatidylcholine, and this entire big chunk here is the phosphatide. So the choline is only this little tiny chunk out at the end. It has a phosphate group, like the terminal end of the choline is charged. This provides for water solubility and makes this compound have emulsion-like properties or detergent-like properties. So this end here, because it doesn't have any charge on it, is very fatty and greasy and oily, and therefore this material is able to connect a fatty material to a water material, so it acts like an emulsion. So it can take 2 things, oil and water, and blend them together. So that's what you find in a lot of foods where lecithin, for example, which is a lot of phosphatidylcholine, is used as an emulsifier to make ice cream and other things like that, where you're mixing fats and waters together. But down here I just put in phosphatidylserine so that you can see 2 different phosphatides and how they're similar and how they're different. Phosphatidylserine, while not being a neurotransmitter, it is a cell signaling factor, so it works like a neurotransmitter, but not in your brain, but in your cells. And it differs because this end here has an amino acid serine on it instead of the dimethylaminoethanol or choline. And again, you see a prevalence of charges. It even has more charge distribution than does phosphatidylcholine. So phosphatidylserine will sit on a membrane, and the charged end will stick out of the membrane. So you can think of it as kind of a ship that floats on the surface, where this is the part that's above the water, and that's the part that's below the water. Yes?
So what about doing IV versus something orally?
Doing IV?
Phosphatidylcholine.
Administering it IV? I couldn't address the technological questions about that. I mean, there is a problem with putting something in IV where it can overload certain systems if it's not handled right. So things need to be in a form that can be assimilated. But I think that lecithin and phosphatidylcholine is part of a lot of different preparations to help with that assimilation. So you can take a fat and mix it with lecithin and then put it in IV and not have little fat globules going around clogging up your arteries and causing circulation problems. But other than that, I can't really address that question. This is acetylcholine again, but this is showing a kind of flow diagram here where we have acetylcholine as a neurotransmitter, and as it's released, this arrow represents the neurotransmission part, which reacts with the acetylcholine receptor, which is in the target tissue, like the ear. And then there's an enzyme called acetylcholinesterase that breaks the acetylcholine back down into choline and acetate. And then it can be recycled again. And this acetylcholinesterase is a rate-limiting enzyme. It's a critical part of the signal because it removes extra acetylcholine. So it cleans the receptor of the previous signal. So it would be like if I'm talking to you and there's an echo machine and the echoes just keep building up and up and up and up and up, eventually you wouldn't be able to hear me. So what this acetylcholinesterase does is it removes the acetylcholine from the synaptic gap such that the next pulse of neurotransmitter can be received as a discrete signal. So when you inhibit this inhibition of acetylcholinesterase, you have malathion pesticides that do this. They basically cause insects to go into a state of muscular seizure where their muscles tighten up and do not relax. Kills the insects. Nerve gas applied to humans. Alzheimer's drugs, because acetylcholine receptors and acetylcholine levels are damaged in that particular neuron class. So by subtly inhibiting that enzyme, one increases the volume of the signal, like using an amplifier. Okay? Huperzine and galantamine are examples of herbal products that do the same kind of thing, where they're mild enough that the chances of you getting a nerve gas reaction are basically non-existent, unless you eat a pound of it or something like that.
Steve?
Yes?
I tried the Huperzine, and I ended up with extremely painful leg cramps. Like, just from one Huperzine.
Yes, so it can be strong, particularly in people for whom the cholinergic nervous system is already dominant. And I'm a good example of that. So this gets to another point that I'll be addressing later, which has to do with the fact that neurotransmitters are not signals that are isolated from each other, but are relative to each other. And so acetylcholine has a balancing neurotransmitter called GABA. Acetylcholine increases muscle tension, GABA reduces muscle tension. And so if your GABA is weak and you stimulate your acetylcholine levels, then that balance is drastically disrupted, which can result in cramps and things like vasospasm, which is like a mini heart attack, only it's temporary. It doesn't permanently block your vessel, but the muscle in the blood vessel just goes and stops blood flow for a while until the muscle gets enough GABA or enough magnesium or whatever to relax again. And then the blood flow resumes. Magnesium, for example, tends to have a balancing effect because it helps relax muscles. And so you have the neurotransmitters that are driving the muscle, then you have electrolytes that are driving the muscle, and if you stack a whole series of factors up that are all on the right side of things, the system will fall over. It's too much out of balance. So this is one of the things that I'll be getting to later, where we talk about some of the complexities involved in figuring this out. So if you want to go out and experiment with your brain, take it out for a drive and see how it works, that you know that this is fundamentally a complicated kind of process. Another neurotransmitter that's involved in the the core of the brain, the emotional or motivational center of the brain, is dopamine. Dopamine is a neurotransmitter of motivation, of drive, of dissatisfaction, an imperative, a biological imperative to make a change in your life. You know, the grass is greener on the other side of the hill because you know it's yellow right here. That kind of thing. This is one diagram that just shows the flow of materials, phenylalanine and tyrosine, and dopa, L-dopa, being amino acids. You find those in your food. The amount of phenylalanine versus tyrosine and dopa will vary from food to food. In typical foods, dopa is a fairly low level nutrient, but in some beans it can be very high. And this is one of the chemical warfare agents that beans use to keep animals from eating them, is by putting very high levels of dopa in it. It makes you very dissatisfied, it increases your dissatisfaction, so you leave the area and you stop eating the beans. This is a pathway, the same pathway, drawn out straight, where it shows in red the enzymes that are involved in the transformations of each of these steps. And on the side here in green, the cofactors that are involved in the transformation. So it's not just that you need the enzymes, you also need copper, iron here, B6 there, copper there, ascorbate, vitamin C, S-adenosylmethionine is a methylating agent. That's composed of methionine and a methyl group and an ATP. Yes?
Is there a way that you know to measure those enzymes in the human body to figure out where that pathway may be blocked?
Yeah, that can be done. That typically can be a problem, though, in terms of what cell population do you look at?
I don't know.
You certainly don't want to do a brain biopsy, right? Right. And red blood cells, which are readily accessible, are not nuclear cells, so they're a very atypical cell population. And so it comes down to how do you look at this? Well, there are fingerprints and byproducts of each one of these reactions where you can pick up certain things in the body. So you can look in urine and see what's happening. If you have the guts to do a spinal tap, you can look in spinal fluid and see what's there. So you can get an idea about what may be going on here. And the other thing that you can do is you can just look for the nutrient. You know, because if there's iron deficiency in one place in the body, there will be iron deficiency in other places of the body, and so it's not likely that this one enzyme will be impaired, but a whole bunch of them are that use the same nutrient. Copper, for example. There's a primary copper deficiency, which is you're not eating enough copper in your food. And then there's a secondary copper deficiency, which is caused by inflammation. You've got, a fever, you've got allergy, your immune system is active, and your body is packing away the copper into storage forms and restricting its bioavailability to the rest of the cells of the body so it doesn't get to be used for a particular enzyme. So you may experience issues where maybe you can't make norepinephrine very well, or maybe the enzyme that produces melanin, it makes you tanned, is not working up to snuff. Or the enzyme that causes collagen maturation, which also uses copper, isn't working well, and so you've got a higher turnover in your collagen. It's not fully matured. So usually these kinds of problems are organism-wide.
Question.
Yes?
So is L-dopa a supplement or a drug?
Both. It's sold by prescription as a drug, and it's also sold as a dietary supplement. You
But
can
it's
buy.
actually a normally occurring body chemical.
Yes, it's found in food, and you can buy it as a supplement, although most of the supplements are actually bean extracts. Yeah. This is to show you the complicated aspects of just one particular factor. It turns out to be a really critical one. Tryptophan is the source for serotonin. And tryptophan is also an amino acid, essential amino acid for making protein. And about 2/3 of the NADH and NADPH that's produced by the body is produced from tryptophan by this convoluted pathway. All of this stuff wandering around here eventually comes down to making this extremely inefficient program. 60 milligrams of tryptophan will make 1 milligram of NAD. That's how inefficient it is. Also, you have on this part here, if you have inflammation, infection, allergy, or fever, a peripheral enzyme is produced called IDO in the bloodstream that's job is to destroy tryptophan and in other indolamines. This particular double ring system here is called an indole ring, and this is an indolamine, that's an indolamine, and that's an indolamine, and this enzyme destroys all of them. So if you have a fever, your level of tryptophan will be lowered. You will not be able to make as much, well, this pathway can be enhanced in the liver, but anything that goes down this pathway never makes it into the cells of your body. So it's like having a hole in your gas tank.
So you shouldn't take tryptophan if you're got a fever.
Right. It's a good idea not to take tryptophan if you have a fever, if you have chronic inflammation, which is why tryptophan works well in people who are healthy or just have at the beginning end of their insomnia. Whereas if it doesn't work very well at all for fibromyalgic patients, for example, because their inflammation is so entrenched that this is a major pathway and has been for, 5, 10, 20 years. Here's a picture of the nerve cell again that shows serotonin production. Dietary protein, maldigestion, and low protein can block this pathway here. Tryptophan supplementation can increase it. You get serum tryptophan in the blood. Catabolism by IDO during fever inflammation can destroy the tryptophan, prevent it from getting across the blood-brain barrier. You have competition from large neutral amino acids, which would be found in protein-rich foods. It's also facilitated by blood glucose and insulin, which would be favored in non-protein meals. So this is a weird thing that when you eat, for example, turkey, which has a lot of tryptophan in it, the turkey doesn't make it into the brain because you have a lot of large neutral amino acids in turkey. So those get through first. It's a first come, first served basis, and the large neutral amino acids are the big, they push onto the train first. And so the brain tryptophan goes down when you eat a high tryptophan food. On the other hand, if you eat, let's say, fruit as a healthy sugar food, or candy as a non-healthy sugar food, you get blood glucose increases in insulin that drop your large neutral amino acid competition, and the tryptophan that's maintained in your blood flows into the brain, so you get a high from sugar. The tryptophan is in the inside the brain, is absorbed into the cell, metabolized into serotonin, excreted. We have the same reuptake inhibitor pathway, and we have postsynaptic response in the cell, just like we did in the other neurotransmitter, and we have the negative feedback loops that control the rate-limiting step that are produced from serotonin, both that comes from its own production inside the cell and from the reuptake and recycling of serotonin back into the cell. So there's the comment I made earlier about SRI drugs. And here's tryptophan and 5-hydroxytryptophan, increase serotonin, but only if they get through the blood-brain barrier. This is an explanation of an example of one neurotransmitter system, the nitric oxide system. This is nitric oxide is drawn as this red diamond shape. And it bonds to its receptor on the cell surface, and then there's another protein called the G protein, which is below it, that extrudes into the cell, and the signal is passed from the receptor to the G protein to an effector, which actually produces something inside the cell. It changes the environment in the cell. So this change in the cell is what the result of the neurotransmitter is. So we have here, a way of modifying nitric oxide from outside, by example, by consuming arginine. Okay? Or we can modify it from the inside by taking Viagra. Viagra blocks this enzyme here, which causes a backup of cyclic GMP. This is the signal, the biological response that's produced. So GTP, this is like ATP, but uses G instead of A. And it's got 3 phosphate groups on it, triphosphate, and 2 of them are removed, and the molecule is like bent around and it fastens to itself in an awkward position. And it's bonded to itself. That's why it's called cyclic GMP. And that fits into a receptor. And then the phosphodiesterase pulls the attached part off, allows the molecule to restore to its original shape. It no longer binds to its biological molecule, and you no longer have a biological response. You now have non-cyclic GMP, which then is phosphorylated back to GTP, so it's available to go back through the cycle again. So this is like an amplifying system. You have your sound waves that are coming into the microphone. They're then going into the electronics where they're being amplified, and by turning down this nozzle, turning this knob, one increases the volume of the signal. Very nice system. All right. Okay, so we have the basic issue of the biochemistry being a property of the brain, and the mind being the property of our consciousness. And so what's the connection between the two? It's certainly not obvious. It certainly doesn't lend itself to science. More of an issue of philosophy. But scientists are looking into how this kind of thing happens, and I can tell you from firsthand experience that by taking neurotransmitter precursors and using drugs to change neurotransmitter levels, you do change your consciousness. You do change your emotions. You do change. I've had people talk to me about switching on, for example, creativity. A woman who was an artist and had 4 kids and completely lost her ability to be creative, and then started taking hydrogen at some point when she was in her late 30s, and immediately started having all these ideas and creative ideas and got back into art again. I can't say I know why that happened, but I do know that it did happen, and I will in a minute be showing you some issues relating to psychological trends and all that I've seen in myself in response to experimenting with the neurotransmitters regulating serotonin versus dopamine. So dopamine, pro-motivational, serotonin calms you down, so they're like an opposite pair. We also have issues of, as I said, creativity, but some people report spiritual issues. There are practices that involve cognitive enhancement like carbon dioxide, which improves blood flow in the brain, in the higher centers of the brain. Chanting and singing and breath holding techniques can enhance carbon dioxide and improve blood flow that change your neurotransmitter levels. We also have genetics. Sex, for example, influences the migration of cells in the brain and the differentiation of cells in the brain. So male brains and female brains are structurally different from each other. They're also, the biochemistry of it is different. We have polymorphisms like affect folic acid metabolism, where people have different types of genes that have different needs for different kinds of nutrients. We have the change that's induced by stresses to the body. So if you have too much potassium in your body, your stress mechanisms will be different, in a different position of up and down regulation than somebody who has too much sodium in their bodies. And then we have anatomical individuality, which has to do with the way your brains develop. So from the period of time when you're born to when you're about 2 years of age, about half the neurons in your brain die. And it's which ones die and which ones live that come to make up you in terms of how your brain is wired. And so there's all kinds of feedback loops in there that reinforce neurons that are wired in particular ways to say, yeah, get stronger, survive this holocaust of neurons, and other neurons that aren't wired correctly where they commit suicide. So this is why children, the first 2 years of a child's life, are absolutely critical to the potential of whether that child will reach. Almost done. This is a diagram of dopamine and serotonin. Dopamine on the left axis, serotonin on the right axis, that has been plotted based on these completely arbitrary levels. Low, normal, high, where we have this quadrant being high for both neurotransmitters, this one being low for both, and how different kinds of experiences and personality traits may correlate with changes in neurotransmitter levels. So, for example, self-assertiveness versus self-transcendence have to do with the relative dominance of serotonin being involved in self-transcendent emotion experience, and the dominance of dopamine involved in self-assertive emotion.
What is self-transcendence?
feeling like you're part of something larger than yourself, appreciating your role in nature, where you see the big picture and you're a part of it. Religious experience, perception of God would be an example of that too. Interpersonal relationships, you know, where the relationship has an importance to you that is way more than just you. On the opposite side of that would be the low end of things. You've got apathy, mild depletion of both dopamine and serotonin, depression, significant depletion, and suicidal depression, which would be an example of severe depletion of those both neurotransmitters. Then if you have out here where you have the dopamine system is way dominant, you have psychosis, an example of an extreme state. And down here, a mild state, obsessiveness, compulsiveness. How many of us have those kinds of personality traits? Some more than others. And Tourette syndrome is a developmental disturbance in the balance of these neurotransmitters caused by too many dopamine receptors of a particular kind. So a smaller amount of dopamine, a normal amount of dopamine, produces a high amount of dopaminergic effect. So I was fortunate enough years ago to run into one of my subscribers who was a translator for the UN. So here's a woman who basically has to function in a highly stressful environment in real time to hear one language coming in and to speak another language out, and she had Tourette syndrome. And she was able to do that on tryptophan by pushing her serotonin level artificially up. She was able to balance out the dopamine and be functional. Without the tryptophan, she couldn't do her job and was disabled. So that's what this can mean to people. So that's an example of a rather extreme state. But all of us have subtle issues with certain neurons and how they're structured in the brain. There are times when neurons in the brain will be growing out and then they'll migrate across the brain. If there's something that happens during that time, let's say your mother gets drunk, and so there's a lot of acetaldehyde that's produced, that can interfere with the migration of those cells and how they wire to each other. And so all of these factors are subject to chaos and random chance and this kind of thing. So it's basically, at this point in our lives, we basically have to accept what's happened up until now and work with what we have. Okay? Any questions?
What does this mean for the psychiatry industry?
Well, first of all, I've observed both psychological and physiological basis for a lot of psychiatric issues. Schizophrenia is something I've been studying for 25 years. And it's my opinion that that there are circumstances where psychological causes are dominant in certain people. But the vast majority of people, it's physiologic. That there's an issue that disturbs metabolism in certain ways, that causes imbalances, that cause dysfunction in certain pathways, that lead to the manifestation of psychiatric symptoms. And I've worked with a dozen schizophrenics over the last 25 years who have become completely functional members of society. You would have no idea that there were times in their lives when they were locked up in wards and were denied any aspect of freedom whatsoever. And the sad thing is that a lot of those people had to employ very extreme measures to get out from underneath those people, including having their families and friends smuggle in nutrient, vitamin supplements, of having people get lawyers in to try to get people out of those kinds of facilities. Because once you've been declared to be unable to make decisions for yourself, it's like, from then on, that they give you neuroleptic drugs, and those produce Parkinson's disease because they imbalance your neurotransmitters and your metabolism in all kinds of ways that the nutrients don't. Any other questions? Yes?
When a message goes down a nerve to your foot, does that have to go through a series of those synapses?
Yes. There are neurons that are very long that go down your spine that are wrapped in insulating materials where one signal can perpetuate over many feet. So it's not like they have to, but there are places where they do branch and adapt. And so a signal will come down to one point and go out a particular way, and that's it's passed from neuron to neuron to neuron like a bucket brigade. The distances involved, in terms of the cellular structure that moves materials down the neurons, not the polarization wave, but the actual infrastructure that delivers, like food, and gasoline and parts and products through the interstate highway system in the United States. That's what we're talking about here. So a neuron, let me just say a dendrite in your brain, an average dendrite in your brain, the mobile system in it is like, if it were equivalent to a 2-lane highway, the dendrite would be the distance between, let's say, Santa Cruz and Maine. 2-lane highway. So if that motor control system that moves material breaks down, the nerve becomes dysfunctional. And that's what a lot of Alzheimer's disease is that the breakdown in the mobile infrastructure, the energy demanding system the gasoline that powers the trucks, that moves the material across the country, the gas supply gets less and less and less until so little material gets moved that the nerve goes into unconsciousness. And that's the good news, because if it's an unconscious neuron, you can wake it up again. If you turn around the metabolism, the neuron will resume its function. It's not like it's dead yet. Now, in situations like Parkinson's disease, there's good evidence that a major amount of the neural damage is permanent. These neurons are just not there anymore. So for those, all you can do is rescue the non-dead cells.
So I'm surprised that all those little exchanges can take place so fast.
Well, you're talking, nanoseconds or even femtoseconds, because the diffusion is that fast. I mean, the distances are so small that it is extremely rapid, and the polarization wave is also very rapid. So that happens because there's the cells have pumps that create electrical gradients. And then there are gates, and the gates become polarized, and the signal switches, and then there's an electromagnetic field that's produced that triggers the next gate and triggers the next gate. And so it's like a series of dominoes that are stacked up, and all you do is tip over the first one, and it goes pump, 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 right down the line. But the first domino isn't triggered by the input signal. So there's like an amplifier or a capacitor or something in the system that means that a certain amount of signaling will then result in the neuron firing. And there may be multiple firings that trigger one firing, or it could be that there's a metabolic change or capacity that's built up in the cell that triggers a firing without much input. So it can work both ways. There's also the fact that certain cells can increase the activity of other cells, and other cells decrease the activity of other cells, so that you have short-term feedback loops and long-term feedback loops where, for example, GHB is a neuromodulator that's associated with GABA. GABA is a muscle relaxation agent, inhibits certain neurons, and the vesicles that store the GABA have some GHB in it. Some of the neurons do, and others don't, and that's released, and that self-modulates the affected neurons and causes other cells in the area to respond to it, where it changes their function. So the GHB will block the release of dopamine, which is why it works to enhance sleep.
Thank you, Lucy. I want to share with you something here that you should all know. I mean, you see what kind of work Steve does. Steve is best. And I want you to know that he also does consultations. I mean, he's probably one of the finest biochemists you're ever going to run into. And with such a detailed and precise knowledge of the way these, especially neurotransmitters, work, and a lot of other things. But he's never gets around to the marketing. I mean, there's a lot of inventors who invent stuff and they never even get around to patenting it. They want to get on to the next invention. There's a little bit of that in Steve. For whatever reasons, I want to tell you that you can call him up and consult with him for $100 an hour and learn things you will never know in any other way. If you know anything about schizophrenia, you know that if you go in an institution, there's 1 in 1,000 that ever is cured. And Steve has had success with schizophrenics. he's had, and that's the toughest, the very toughest condition I think there is in the field of
No.
psychiatry.
Parkinson's disease is the toughest.
Okay, okay. Well, you know, Parkinson's maybe because it just kills the neurons. But anyway, what I'm telling you is this. If you have a problem, or if you have a friend who has a problem, and it doesn't have to be extreme problem, it could be, you know, just you're not what you wanted to be, you're not what you're used to being, call Steve up and get together with him. Information isn't going to hurt you any. It's going to give you options. And I feel like that I, as you can tell, I feel very strong. I want to promote his service, not because I want to see him get rich, because God knows he'll never get rich because he's not interested in money, and it just doesn't seem to follow him around. But because I want to see you all get access to this knowledge that is so rare and so precious. So please make a note of that. That's how you, whoops, not there anymore. If you have any trouble reaching him, call me and I will put you in touch with him personally. Steve Fowkes, F-A-W-K-E-S. And what, you want to give a phone number, Steve, again?
650-321-2374.
Okay. 650-321-6574.
2374.
So, sorry. 23.
C-E-R-I. on
Yeah,
your phone.
2374. Okay.
And the website is www.ceri. So the phone number and the website are the same. CERI.
And it's just, Steve is just a remarkable and precious resource. And as you can tell, I mean, I personally am very indebted to him for turning me on to Depronel, which is a drug that practically everybody over 45 should be taking, because it increases dopamine, and it gave me back the kind of energy and motivation that I was used to having, so that I can, get some things done for the Smart Life Forum and a few other things, and also continue to enjoy might say a blessed sex life at 67. So, which was not there 10 years ago before I started taking Depronel. So, I mean, this is important stuff. I mean, if you love life, you got to keep your dopamine up. So anyway, okay, I don't want to carry on. We've got a great speaker coming up, but I do want to take a 5-minute break. So everybody get up, stretch, take some deep breaths, and actually make it a 3-minute break. At 10 after, we'll have Mike Corrick introduce our main speaker.